This protocol describes how to culture and exposure human bronchial epithelial cells via the air. This is a more realistic model for studying exposure of the lung to inhaled particles. The lung cells prepared with this method resemble the real environment in the lung. The cells will produce mucus and form a tight barrier, remaining viable for several weeks.
This method can be used to study toxic effects of airborne particles. It can also be used to study infectious diseases of the airways and drugs taken by inhalation. When attempting this protocol for the first time, keep in mind that generating an aerosol requires specialist knowledge. It also requires dedicated equipment for measurement.
Begin by diluting the cells in pre-warmed cell culture medium, or CCM, to a concentration of 500,000 cells per milliliter for 6-well inserts, and 250,000 cells per milliliter for 12-well inserts. Place a cell culture plate with inserts under aseptic conditions, and fill the basolateral side with pre-warmed CCM. After mixing the cell suspension, pipette it on top of the membrane in the cell culture insert. Cover the plate and incubate it at 37 degrees Celsius and 5% carbon dioxide. Culture the cells under submerged conditions for 7 days to allow them to reach confluency, changing the CCM every 2 to 3 days. To measure the trans-epithelial electrical resistance, use a charged epithelial voltometer supplemented with a chopstick electrode set. Clean the electrodes with 70% ethanol.